Hey folks, welcome to another edition of Zog Science. Today we're going to be taking a look at how cells transport things in and out through the cell membrane. So let's talk about the basics to begin with. It's kind of some background information. Matter wants to spread out, and that's a consequence of the second law of thermodynamics, which states that the entropy, which is another word for disorder of a system, will increase unless energy is added back into that system. So basically, on its own, all matter will spread out. This is something that is natural. You think about like a kitchen that starts smelling and then the whole house starts to smell. That's because the molecules that create that smell started off in the kitchen, but they are slowly spreading out throughout the rest of the house. Another example is your room. Your room starts out at a very ordered state. However, unless you continue to put effort and energy into keeping your room ordered, it will eventually go towards disorder. Now, when we talk about this idea of thermodynamics, we also know that energy cannot be created nor destroyed. But that's on a global and kind of a universal scale. On a very small scale, energy is constantly being transferred in and out of a system. So it is sometimes difficult to see that. Let me get that back. There we go. Sometimes it's kind of hard to think about the whole idea that energy isn't created or destroyed when we talk about individual cells, but on a universal scale, that is the law, and that's what follows that. So passive transport. Passive transport is basically when there is no energy being added into a system, and so therefore materials are going to move from areas of high concentration to low concentration. Those brackets denote concentration. Now, the reason why this occurs is that when you have a higher concentration of something, you're going to have those molecules running into each other more often, which means that they're going to want to push out. There's kind of like a little bit of pressure that forms, so they're going to want to push out. So they're going to go from an area where there are a lot of the molecules hitting to an area where they are evenly hitting. They're equally spread out. There are three different types of passive transport. You've got diffusion, facilitated diffusion, and osmosis. And passive transport occurs spontaneously all the time, so long as you can pass through the cell membrane. So some of the time the membrane the cell membrane is selectively permeable, so not all materials can pass through it. So only those materials that can actually pass through the membrane will be involved in the actual passive transport process. So let's talk about diffusion. Diffusion is just the movement of molecules across the membrane. And again, their goal is to reach equilibrium. Equilibrium is not a static state. Rather, it is a state where you have an equal concentration of molecules on each side. You know, molecules are kind of moving back and forth across it at all times. You can kind of see a couple of examples here. You've got your molecules moving back and forth to really reach equilibrium. You've got the same concentration on both sides. Here you've got different molecules on either side. You've got diffusion occurring until they both reach equilibrium. We also can have facilitated diffusion. So recall that the cell membrane is not permeable except for to some small nonpolar molecules and a few ions. But we do have, fortunately, some transport proteins that can help small polar molecules and other charged particles cross the membrane. There are kind of two different types. We've got channel proteins and carrier proteins. Both of them work spontaneously, so you don't have to add energy to those proteins in order for them to function. They will just function on their own. And then the last example of passive transport is osmosis. Osmosis is only different from diffusion, and that is specifically the movement of water molecules in order to reach equilibrium. And water molecules tend to move when you have another molecule that cannot move past the membrane. So, for example, here we've got a selectively permeable membrane that is not allowing the sugar molecules to pass through. So the water is going to move so that now there is an equal concentration. Notice that that's different than saying that there's an equal amount on either side, right? Instead, what we're saying is that there's going to be an equal concentration of sugar molecules, or in other words, another equal ratio of sugar to water molecules on both sides of the membrane. Now, when we talk about water moving around, we're going to run into this concept of tonicity, which is the ability of surrounding solutions to cause a cell to gain or lose water. And there are kind of three different tonicities that we'll discuss. We've got hypotonic, which means there is more water outside the cell. If there is more water outside the cell, the water is going to go into the cell. And if it is an animal cell, that will cause the cell to lyse or break open because the water is going to fill it up to the point where the cell will burst. The cell membrane will actually burst. We also can have, in a plant cell, this is actually what you want. So this is part of the reason why you want your plants to be well watered, is that you need a lot of water going into the cell. The cell wall will hold the structure of the cell, and you have what is known as a turgid cell, which is when a plant is nice and crisp and upright. We can also have a hypertonic solution. Here we've got more water inside the cell than outside the cell, so the water leaves the cell, causing the cell to shrivel up. And this is when your plants are kind of wilting, right? This is what is going on. They're not getting enough water. And then we've got an isotonic solution, which is where we have an equilibrium. For plants, this isn't the best, but it's also not terrible. They're not completely, falling over, but they probably are a little limp on the side. So let's take a look at an example. So in this case, our cell is permeable to glucose and fructose, but not sucrose. So let's think about this. What's going to happen? Well, the sucrose is not going to be able to move, right? Which means that our water is going to probably be moving around. The glucose has a higher concentration inside than outside. So our glucose is going to move from the cell out of the cell. We have fructose outside the cell, but not inside the cell. So the fructose is going to go in. So here's what that looks like. The water is going to go into the cell in order to balance out the molarities of the sucrose. The glucose is going to leave, and the fructose is going to go in. We can also have, when we're dealing with water regulation, specific organelles that are built for this. And this is a what is known as a contractile vacuole. So what happens is that the vacuole fills with water and other ions inside the cell, and then it will expel that water out. So basically, it allows the cell to live in an environment where it is hypotonic, in a hypotonic environment without bursting, because it's able to push water back outside of itself. Active transport, okay? So passive transport, remember, these are all involving equilibrium. They're going to be movement of water molecules without energy. Active transport, we're going to have to use energy or ATP in order to move molecules against the concentration gradient or from a low to high concentration. It does require that energy again, and some sort of protein complex is going to have to be involved because we're going to need something to actually do the movement of the molecule. So a couple of examples. Here we've got energy that is going to be moving a proton across here. We will see this a lot more when we get into the mitochondria and we start looking at cellular respiration at a deeper level. We'll see a lot of proton pumps being involved. And here we can actually have two pumps being involved. So what happens is that the ATP pumps the proton outside. The proton then drives this sucrose co-transporter, bringing the proton back inside where it can then be pumped outside again, which enables the cell to move sucrose into the cell, which is obviously a good thing. Another really classic example of a protein pump is the potassium-sodium pump. This is definitely one that you want to be aware of how it works. Basically what happens is that in your nerve cells, you have a high concentration of sodium or potassium inside the cell and a high concentration of sodium outside the cell. And in order to drive the action potential of a nerve, you need to maintain that concentration. So what happens is that we have a protein that fills up with sodium. Then we have an ATP that comes along. This phosphorylation word, that's just where the phosphate group from the ATP breaks off and it forms a phosphate and an ADP. The phosphate then, once it is bound with the protein, causes a conformational change or a structural change that then releases the sodium outside the cell. When it does that, the protein has changed shapes so that now the potassium that is outside the cell can bind. The phosphate group then gets kicked off. The potassium is then released back inside the cell, and the cycle can continue. Cells also will sometimes need to move larger things than just single molecules or single ions. We have this idea of, or this concept of endocytosis. There are 3 different endocytosis, or types of endocytosis, I guess. We've got phagocytosis, is where we have food or other kind of really large particles that are brought in. You notice that the cell membrane kind of wraps around and then pinches off, forming a food vacuole. Next, we have penocytosis. This is where we're just kind of bringing in small molecules, maybe water with some other solute in it, and just for a vesicle is going to be formed, which is going to be transported around. And then we can have receptor-mediated endocytosis. Here there is kind of a protein receptor that's on the outside of the cell, and when that protein receptor gets filled up with molecules, that will then actually bring in the protein or the molecules that are outside the cell in. So it's not going to just be happening kind of on its own, but rather it's going to be kind of forced to happen when you have specific things. So this kind of allows the cell to be a little bit pickier in terms of how it actually moves things in and out, because it's not just randomly engulfing things. It can kind of look for specific molecules. And then we've got exocytosis. Exocytosis is where, again, we talked about this before a little bit with the Golgi apparatus. Then we'll send out molecules, proteins, whatever it is. It can then release that into the extracellular fluid to then go to other cells and other parts of the organism. All right, that's all I've got for you today. Thanks for watching, and I'll see you next time.